Welcome to this lecture on the basics of neuron classification. The human brain is one of the most complex structures in the known universe, and to understand how it works, we first need to understand its building blocks. By the end of this video, you will know exactly how scientists categorize these incredible cells and why this classification is absolutely vital for both basic biology and modern medicine. Let us begin our journey into the microscopic world of the nervous system. To start, I want you to imagine a massive, bustling city. In this city, billions of workers are collaborating to keep everything running smoothly. Some are long-distance truck drivers, others are local delivery riders, and some are managers processing information. If you wanted to truly understand how this city functions, you would not just say they are all workers. You would classify them by their specific tools and jobs. The human brain is exactly like this complex city, containing roughly 86 billion workers called neurons. To understand how we think, feel, and move, we must first classify these neurons into distinct categories based on their unique shapes and roles. Now that we understand why we need to classify neurons, let us look at how we categorize them by their function. Functionally, neurons are divided into three main groups based on the direction of information flow. First, we have sensory neurons, which act like the city's scouts, bringing information from the outside world into the brain. Second, we have motor neurons, which act as the dispatchers, sending movement commands out to our muscles. Finally, we have interneurons. These are the local connectors that link other neurons together in incredibly complex webs, allowing for advanced processing and decision-making. This functional division is our first major step in mapping the nervous system. Beyond their functional roles, we can also classify neurons by the language they speak. Neurons do not physically touch each other. Instead, they communicate across tiny, microscopic gaps known as synapses. To send a message across this gap, they release specific chemical messengers called neurotransmitters. Because different neurons produce and release different types of neurotransmitters, scientists can classify them based on this chemical signature. By grouping neurons into these distinct, well-defined chemical classes, neuroscientists can accurately map out the incredibly complex wiring diagrams of the entire nervous system, understanding exactly which chemical signals are driving our thoughts and behaviors. This brings us to an important question. Why does classifying neurons matter so much in the real world of medicine? Imagine trying to fix a broken computer without knowing the difference between a memory chip and a power supply. It would be nearly impossible. In medicine, different neurological diseases attack very specific types of neurons, while leaving others completely unharmed. For example, amyotrophic lateral sclerosis, or ALS, specifically destroys motor neurons, leaving a patient's sensory neurons completely untouched. Similarly, Parkinson's disease specifically targets a tiny population of neurons that produce the neurotransmitter dopamine. By knowing exactly which class of neuron is failing, doctors can design highly targeted drugs to treat these devastating conditions. Having seen the medical importance of classification, let us now focus on how we classify neurons structurally. Structural classification is the systematic categorization of nerve cells based on their specific anatomical characteristics. When we look at a neuron under a microscope, we see a main cell body, which is known as the soma. Extending outward from this soma are physical projections called neurites. These neurites act as the biological wires of the cell and include both the axons, which send signals away, and the dendrites, which receive incoming signals. By simply counting the number of these projections, we can create strict, objective rules for categorizing cells without any human bias. To make our structural classification completely objective, we can represent it mathematically. Although neuron classification is primarily a biological concept, this mathematical abstraction helps us create strict rules. The formula is simple. N total equals N axon plus N dendrites. Here, N total represents the total number of neurites extending from the cell body. N axon represents the number of axons, which are the long cables that send electrical signals away from the cell. Finally, N dendrites represents the number of dendrites, which are the branching, tree-like structures that receive incoming signals. If the number of dendrites increases, the neuron's ability to receive and integrate inputs from many different sources increases dramatically. Now that we have our mathematical formula for counting neurites, let us establish the rules for categorizing them. Based on the final value of N total, we classify the neuron into specific structural categories. If N total equals exactly 1, the neuron is classified as unipolar. If N total equals exactly 2, it is classified as a bipolar neuron. And if N total is greater than or equal to 3, it is classified as a multipolar neuron. As you can see, this simple numerical threshold system removes all guesswork. A scientist in one lab will classify a cell the exact same way as a scientist in another lab, ensuring consistency across all of neuroscience. Before we move on, let us see this formula in action with a concrete example. A researcher is examining a pyramidal neuron in the cerebral cortex. She observes exactly 1 axon extending downward and exactly 7 distinct dendritic branches extending outward. We are asked to calculate the total number of neurites and classify the cell. First, we identify our given values. N axon is 1 and N dendrites is 7. We plug these into our summation equation, adding the single output cable to the 7 input cables. This yields a total of 8 neurites. Because 8 is greater than or equal to 3, we definitively classify this as a multipolar neuron. This makes perfect physical sense, as cortical neurons must integrate thousands of inputs to process complex thoughts. Let us reinforce this with a second example. This time, looking at a specialized cell deep within the human retina. An ophthalmologist notes this neuron has exactly 1 dendrite receiving light signals and exactly 1 axon transmitting that signal deeper into the eye. We need to classify this neuron. We identify our inputs. N axon is 1 and N dendrites is 1. Substituting these into our formula, we get 1 plus 1, which equals 2. According to our classification rules, a neuron with exactly 2 projections is a bipolar neuron. This highly linear structure is characteristic of sensory pathways like vision, where signals must be passed forward quickly and accurately without being mixed with distracting inputs. Now that we have calculated these classifications mathematically, let us look at what they actually look like physically. On screen is a diagram comparing the 3 structural types we just discussed. On the far left, you see a unipolar neuron, where only a single process emerges from the cell body before splitting. In the middle is the bipolar neuron, just like the retinal cell from our example, with exactly 2 distinct extensions, 1 dendrite and 1 axon, reaching out from opposite sides of the soma. Finally, on the right is the multipolar neuron. Notice the single axon pointing downward, but the massive tree-like canopy of multiple dendrites surrounding the cell body.
This visual perfectly illustrates how structure dictates the neuron's ability to receive and process information.
We have covered a lot of ground today, so let us summarize our key takeaways. First, we learned that neuron classification is the essential first step in decoding the immense complexity of the nervous system, categorizing cells by their structure, function, and chemical signatures. Second, we saw that structural classification is highly objective, relying on a simple mathematical formula that counts the total number of neurites, both axons and dendrites. Finally, we discovered that whether a neuron is a simple bipolar sensory cell or a massive multipolar processing cell, its specific classification dictates exactly how it contributes to our survival. Mastering these classifications allows us to better understand both the healthy brain and the diseases that afflict it. Great job following along with this fundamental topic in neuroscience.